All right, in Chapter 9, we're going to take a look at microbial growth. So we're going to take a look at how microorganisms grow, what they require in order to grow, and the different effects of things like pH and temperature on growth. So first, let's take a look at how microbes grow here in 9.1. So our bacterial cell cycle involves the formation of new cells through the replication of DNA and then partitioning of cellular components into two daughter cells. So this is reminiscent of mitosis in human cells or eukaryotic cells, where we go through this process of making copies of all of the things in the cell and then going through cytokinesis, which is separating those things into two separate sections, squeezing the middle, and then making it go into two different sections, and then ultimately pinching it and making it be two different cells. So we do see this in prokaryotes, except it's slightly different because we don't go through meiosis, which happens when we make daughter cells or when we make new human beings, versus in prokaryotes, in bacteria, they don't have meiosis. They do a mitosis type situation, which is actually called binary fission. So we'll take a closer look at that. In prokaryotes, reproduction is always asexual. Although extensive genetic recombination in the form of horizontal gene transfer takes place. So we do see genes moving back and forth horizontally, meaning between different cells. So that may end up having a different type of daughter cell as compared to the original cell that started, but that is different. It's not actually like a meiosis situation where we are adjusting the DNA, changing the DNA, and then mixing the DNA with another organism's DNA to make a brand new being, in the case of humans. But in this case, we're going to have asexual reproduction, and it's just a division and an exact copy of the original, with the exception of this horizontal gene transfer that can happen sometimes. And we're going to take a look at that in a future chapter. So most bacteria have a single circular chromosome. There are some exceptions, of course, like Borrelia burgdorferi. The Lyme disease bacteria has a linear chromosome. So let's take a look at our typical steps of binary fission. So the most common mechanism of cell replication in bacteria is what is called binary fission. So this is in 9.2, which we'll take a look at in a minute. But before dividing, the cell is going to grow and then increase its number of cellular components. So the cell is going to initiate this binary fission by going through the process of making copies of all of its cellular components. So different proteins that it has, different enzymes it has, it's going to basically double plus some, in some cases, all of its components, everything except for the DNA that we're talking about right now. So it's going to make copies of all of this stuff, it's going to make more cytoplasm, and then it's going to get to the DNA piece. Once it's made lots of copies of all of the stuff within the cell, with the exception of the DNA, then we go to the DNA. So then the replication of DNA starts, and this starts at a location on the chromosome called the origin. So the origin of replication. And then this is also where the chromosome is attached to the inner cell membrane. So a piece or a part of the chromosome, this circular chromosome, is going to be attached to the cell, so it's not really floating around all that freely. And it's at that point, called the origin of replication, where we're going to start this replication process and make a copy of the DNA. So then replication is going to continue, and if you don't remember replication specifically, you can look at the previous chapter. In Chapter 8 it talks about replication, but also you should remember that from previous courses, how genetic replication happens. So we have our origin of replication, and then our replication is going to continue in opposite directions along the chromosome. So we have a circular chromosome. It's going to start in one position, in one spot called the origin. It's going to go both directions. So opposite directions along the chromosome until the terminus is reached. So until we get around and we reach the terminus, which is terminating the replication. And then we have a whole separate but same chromosome. Now we have two chromosomes that are the same within this single cell, which isn't a large cell. It has multiple copies of all of its components. So the center of the enlarged cell is then going to constrict. So the center of this big cell is then going to start pulling in toward the center. And then it's going to do that until two daughter cells are formed. So it's going to pull in so much that it's eventually going to kind of pinch the middle and then pinch the two cells apart. So each offspring is going to receive a complete copy of the parental genome and a division of the cytoplasm. So they're going to get the cytoplasm, they're going to get an exact copy, both of them will have a complete copy of the chromosome, and then they'll have all of the cellular components as well. So don't forget about those pieces that have been duplicated. Those cellular components are going to go into each cell so that each cell is an exact copy. And this last piece here, where it divides up the cytoplasm, is called cytokinesis. So cytoplasm and kinesis, so the moving of the cytoplasm. So this process of cytokinesis and cell division is directed by this protein called FTSZ. So this FTSZ is what's actually going to create this kind of pinching off part. The FTSZ is going to assemble into a Z ring on the cytoplasmic membrane. And then this Z ring is going to be anchored to the cell by FTSZ binding proteins. And then this position is going to define the division plane between the two daughter cells. So this FTSZ protein is going to assemble into a Z ring, so a ring kind of stretching around, it's within the cell, but going the length of the cell, the circumference of the cell, and then it's going to be anchored to the actual cell by these FTSZ binding proteins. And then what's going to happen is that we are going to have additional proteins come. So additional proteins are going to be added to the Z ring, and then it's going to then be called a divisome. So we have this Z ring which has formed, it anchors itself, and then it's going to have additional proteins come along to assist it, and then that's called the divisome. And then this divisome is going to be activated. It's going to activate to produce peptidoglycan cell wall and build a septum, and that's going to divide the two daughter cells. So this Z ring plus these other proteins altogether called the divisome is going to activate, and what's going to happen is it's going to pull in on the edges of the cell, on that membrane. It's going to produce a peptidoglycan cell wall, so it's going to help to build an additional cell wall between the two parts of the cell, and going to build the septum. So the septum is this division between the two cells, and we're going to look at the image in just a minute. So the daughter cells are separated by this division septum, where all of the cell's outer layers, meaning the cell wall and the outer membranes, if all of these things exist, meaning the cell wall and the outer membranes, are then remodeled to complete the division process. So here, if we look at the image now in B, in this first figure here, we see the simplified form of binary fission. So we see our kind of enlarged cell here with our DNA, our circular chromosome right here, and then the red here is indicating our origin of replication. So then we go through the process of DNA replication. So we start at the origin, we go both directions, so we go this direction and this direction all the way around until we get to the terminus. Then we have exact copies of their circular chromosome, their DNA, inside of this elongated cell. You can see it's even grown even larger here. Then we have the formation of this division septum, or the septum. And what we see here, I can scroll down just a little bit more so we can see both. But down here, we see that the cytokinesis piece of it. So we see that we have what's called a cleavage furrow, which is this kind of pulling inward of the outer membranes, so the plasma membrane and the cell wall, pulling that in toward the center. And this is due to this FTSZ ring. So you can see they've kind of placed these proteins, this Z ring, and its additional proteins in this center area. So it's kind of going along this region here and then back. Hard to draw three-dimensionally, but and then it's going to be pulling this down in toward it and pulling this piece down into it. So we have a cleavage furrow, and then we see here that's pulling down in, and then we have the formation of the septum. Once we have the septum, we have cell separation. So now we have two separate cells. So if we scroll down, we can see the rest of this figure here where you can really see the septum. So we have our FTSZ ring. Remember, it's pulling in, kind of like a rubber band. So it's placed on there and it kind of pulls tighter. So I guess like a rubber band is pulling in this middle of this elongated cell. But if we could then take the rubber band, for example, and pull it tighter and tighter and tighter, like a drawstring. So we have a drawstring and we're pulling it tighter and tighter and tighter. And as we're doing this, you can see right here is our Z ring. And that Z ring is helping to create this septum. And the septum is composed of the plasma membrane as well as the cell wall. So it's building all of those pieces there. And then what we see is then once this septum has been completed, then we are able to separate the two, because now we have plasma membrane on both sides, and then we have cell wall on both sides, and then it ends up pinching off completely, and now we have two separate cells. All right. So then next we're going to take a look at generation time. So in a eukaryotic organism, our generation time, so for example in humans, is the time between the same points of the life cycle in two successive generations. So if we talk about, so for example, it says the typical generation time for the human population is 25 years. So 25 years would be, for example, our first generation, let's say the first generation being born, like say when you were born, and then the next generation would be if you had a child when that child was born. And so the typical generation time for the human population is about 25 years. Basically, the way we would say that, rather than saying our generation time for a human population is 25 years, we would likely say people generally have children when they're about 25 years old, which is the same thing, our generation time. However, this definition doesn't really work for bacteria or archaea, because they reproduce very rapidly. So many of them reproduce so rapidly that a generation time would be seconds. Also, others remain dormant for thousands of years. So you can't really say that that organism, for example, has a generation time of thousands and thousands of years. It just happens to be dormant at that time. But then once it goes through the process of making generations, then it may be much faster than that. So instead, what we do is in prokaryotes, so bacteria and archaea, the generation time is also called the doubling time. So it's defined as the time it takes for the population to double through one round of binary fission. So basically, when we double the population, so we can start with 10 cells, once we get 20 cells, then that's the doubling time or the generation time. If we start with 100,000 cells, when we get 200,000 cells, that's our doubling time. So bacterial doubling times vary enormously. So, for example, E. coli can double in as little as 20 minutes under optimal growth conditions, like in a laboratory. Bacteria of the same species may need several days to double, especially in harsh environments. So if we plated some E. coli and we put it in an incubator at 37 degrees, which is when it's happy, it'll go through binary fission in about 20 minutes. So we'll have our generation time at about 20 minutes. But, for example, if we took E. coli and we put it in a refrigerator, or if we put it in some intense heat, an example of harsh environments, then it may take much, much longer. For example, several days if we put it in a refrigerator, for example. Most pathogens grow rapidly, like E. coli, so 20 minutes, very fast, and most of them are fast like that. But of course there are exceptions to that. So for example, Mycobacterium tuberculosis has a generation time of between 15 and 20 hours. So the incubation time on that, if you were trying to grow Mycobacterium tuberculosis, would be much longer, versus if you were working with an E. coli species, then you could get through this generation time fairly quickly and move on with the experiments very quickly. On the other hand, we have Mycobacterium leprae, which causes leprosy, grows much more slowly. So its doubling time is 14 days. So even in optimum conditions, if we were working in a laboratory with this organism, for example, then that would take much, much longer to go through that process. So that is our generation time.